sensitive, then you use immune suppressants and anti-inflammatories. And of course, the problem with that is that you leave yourself open for infection in general, right? And that's just, you pick your poison basically, right? We don't want to do that. So when we're talking about these types of conditions, all right, your immune system normally is supposed to do what? It's supposed to recognize foreign antigen, okay, and leave your own antigens alone, all right, which means that anything that comes in bearing a surface molecule other than what you have on your own tissues is a target for destruction, okay? When we overreact to that, all right, and produce massive release of cytokines and histamine, we get systemic inflammation, okay, and that's an example of stuff that you see in things like septic shock or anaphylactic shock, okay, and the problem there, of course, is all your blood vessels open up and your blood pressure drops so quick that you can go into a coma and die, all right. So how do we deal with something like that? Well, we give you immune suppressants and anti-inflammatories, all right, to deal with the hypersensitivity. Um, there's four types of hypersensitivity. We'll go into the numbered one, two, three, and four, right? And then autoimmunity is simply an attack on your own tissue. And the theory as to how that happens is varied, okay? Um, one theory is that there's a mutation in either the immune system or your own tissue, and that initiates an attack on your own tissue, either because you make an antibody or T cell receptor that you didn't make before, or because you make a surface molecule that you didn't make before. Okay. Another theory is that when your T cell population is being produced in your thymus, the cells that don't pass the test of being able to tolerate your own antigens and attack other antigens are either destroyed or they're rendered in a state of what's called anergy, Anergy. And for reasons that we don't fully understand, sometimes those cells that are in anergy wake up and cause trouble. Okay. So that's another possibility, right? The other possibility is breaking contain between an immunologically privileged compartment and your immune system, such as in the central nervous system. Okay. This, for instance, is the mechanism of multiple sclerosis, right? Where the blood-brain barrier is breached somehow, either through trauma or through some disease process. And the immune system sneaks in there after the pathogen, and it's never seen stuff like myelin before. And so it tears up the myelin in the upper motor neurons. And as a result, it's difficult for you to pass motor commands from the frontal lobe of your brain down to the lower motor neurons where you can initiate muscle activity. Okay. And it's generally a progressive disorder, and the muscles lose muscle tone and eventually they atrophy. Okay. And then the other mechanism is one that's probably more prevalent than we realize, which is exposure to a cross-reacting antigen. All right. So somehow in your activities of daily living, you come across an antigen that's similar but not identical to your own, or an antigen that gets presented on the surface of a cell along with your own antigens somehow. Okay. And you select then for lymphocytes that recognize that display. And after they clear the antigen, they turn around and they attack your own tissue. Okay. Through the production of autoantibodies and the production of cytotoxic Ts that go after tissues that are displaying something that they think is alien. Okay. So those are all major problems because we have to deal with them by suppressing the entire immune system, and that leaves you open for infection. Right? Now, the other class of immune disorders is immune deficiency. Okay. And immune deficiency can either be acquired or inherited. Okay. Acquired immune deficiency would be stuff like chemo and radiation, treatment for cancer, or AIDS. Okay. Um, Inherited immune deficiency would be stuff like DeGeorge syndrome, A gamma globulinemia. Okay. It's just, or SCIDS, which is basically you're
Excuse
born with
me.
no immune system at all. Right. And the result
Hello,
of that,
Mr. Richard.
yes, ma'am.
There's someone that says she did not receive any link from you.
Oh, it should be the same link as before.
Yeah, she said, I'm trying to tell her, but it seems like she can't find anything.
Can you want to email her the link?
Okay.
Yeah, just do that. Um, Everybody should be good to go. Um, but anyway, in SCIDS, you're born without any immune system at all, right? So you have to live in a sterile environment your entire life. Okay. The fix for stuff like that is to replace your bone marrow. All right. That's very dangerous because your red bone marrow, which is primarily in your axial skeleton and in the proximal heads of your humerus and your femur, produces all your blood, right? And if we replace your bone marrow and it doesn't take, you're going to die. Okay. So that's sort of a last ditch effort to try and fix the problem. All right. So when we look at factors for infection, right, infectious disease, communicability is an important aspect to consider, right? How quickly does it spread? How does it spread? Is it airborne? Is it fecal oral? How does it go from one individual to another? The ability to spread from an individual to others and cause disease is communicability. Measles and pertussis spread easily. HIV, less so. Infectivity is just the ability of a pathogen to invade and multiply in the host. It involves the attachment to cell surface, the release of enzyme, escape of phagocytes, spread through the lymph and the blood and to tissues, right? And virulence is nothing more than the ability to cause disease, okay? And and the virulence of something is also sometimes termed its pathogenicity, right? Something that's extremely pathogenic requires a very low infectious dose to produce disease, okay? Something that's much less pathogenic requires a much higher infectious dose, okay? So rickettsia is an example of an extremely infectious disease. It only takes one cell to produce the condition, okay? Whereas cholera takes around 1 million, okay? So cholera is much less pathogenic than rickettsia, all right? The port of entry is important, right? In order to be infected, the pathogen has to come in the right way, okay? It can be through direct contact, it can be inhaled, ingested, and it can be through the bite of an animal or an insect, okay? But if the pathogen comes in the wrong portal, then it's not going to make you sick, right? For instance, if you get, I don't know, RSV, respiratory syncytial virus, on your skin, all right, you're not going to have RSV infection, right? It has to come in through the nose or the mouth, right? Be inhaled or ingested, okay? You also have to have had no previous exposure to the organism, which means you're going to evoke what's called a primary immune response, okay? Which takes longer to ramp up, which means you're going to be sick for a longer period of time. Hold on. Oh, all right. All right. So that's critical, right? If you've been already exposed to the pathogen, right, and you get a secondary exposure to the pathogen, we already know from adaptive immunity that you're likely not going to get sick unless it's a strain that you haven't encountered before. Toxigenicity is just the ability to produce soluble toxins or endotoxins, which are factors that greatly influence the pathogen's degree of virulence. Okay, so an endotoxin will be something like lipopolysaccharide, which is part of the gram-negative cell wall. All right, and that generally gets released when the organism is destroyed and it comes off in little fragments, and then your body mounts an inflammatory response to it. Okay, an exotoxin would be something like botulism toxin or tetanus toxin, which is excreted by the organism and exerts its effect independent of where the cell is. Okay. All right. So bacteria are prokaryotes, meaning what? They don't have a nuclear membrane around their genetic material. And these were probably the first organisms on the planet. All right. The archaea came first, the bacteria came later. Okay. And they're probably the most numerous organisms on the planet. All right. They're the most numerous organisms on and in your body, too. Okay. They can be aerobic or anaerobic. Okay. Aerobic means that they require oxygen in order to survive. Anaerobic means that they need to be away from oxygen to survive. Now, actually, we can split this into some subcategories, all right, which you should be aware of. So let's pick that up. Okay. Aerobic or anaerobic, we can call these, obligate aerobes. Okay. So you call them facultative. Okay. Or we can call these obligate anaerobes. All right. So what do each of those phrases mean? An obligate aerobe means it has to have oxygen to survive. Cannot survive without it. Okay. A facultative organism can do either high or low or absent oxygen. And so it's a switch hitter. Okay. And an obligate anaerobe is has to stay away from oxygen or oxygen will kill it. Okay. So those are the subcategories, right? And then an organism can be gram positive or gram negative, or it can be acid fast. Okay. Um, Gram staining is one of the ways that we classify bacteria by subjecting them to a differential stain, which is what the gram staining procedure is. Okay. We, at the end of the procedure, have cells that are either purple or pink. Okay. A pink cell is going to be gram negative, a purple cell is going to be gram positive, and that tells us something about uh, what sort of organism it is. Okay. These are some of the classic ways that we identify microbes through phenotypic characteristics, right? Physical characteristics that are observable in a microscope or on a culture plate, okay, or in a chemical reaction. Some organisms don't stain with the gram stain. They're either acid fast or they're not acid fast. If they're acid fast, they end up red, and if they're non-acid fast, they end up blue at the end of the procedure. Okay, and so they have a different type of outer coating than the gram positives and the gram negatives. So there's all sorts of differential stains that you can use to try and figure out what you're working with. Okay. An example, right? Staph aureus, which lives on your skin, your mucous membranes, right? It can be life-threatening. It's a major cause of nosocomial infections in the form of something called MRSA, MRSA, okay, which is an acronym that stands for methicillin-resistant Staph aureus, okay? It's opportunistic, okay, meaning it likes to take advantage of a weakened immune system. It can exist in biofilms, right? We already know biofilms are communities of different microbes living in a matrix that are extremely difficult to kill because of the presence of the matrix and the fact that they express genes inside the biofilm that they don't express when they're out there on their own. An example of a biofilm would be the plaque on your teeth every morning before you brush it. Okay. It secretes exotoxins. These would be things like enzymes that can digest through connective tissue, like hyaluronidase. Okay. So it can chew its way to its destination. That's a virulence factor, right? And antibiotic resistance is a major problem. Methicillin, like in methicillin resistant Staph aureus, is a derivative of penicillin that was created because penicillin is easily destroyed by lots of pathogenic bacteria, right? But as with any organism that has genetic material in it, once you start using a treatment to fight it, you're going to select for organisms in the population that are resistant to the treatment. That goes for viruses, bacteria, protozoans, helminths, you name it. Okay. So what's happened in clinical settings is that because of the use of methicillin, we've selected for methicillin-resistant organisms. So now we can't use the penicillin class on that type of Staph aureus anymore. And this is one of the major sources of nosocomial infection. Now, the other thing to understand about bacterial diseases and bacterial virulence factors is that bacteria can pick up genetic material in a variety of ways, right? They can do it through conjugation. They can do it through transduction. Transposition, or transformation. And you might say, well, what's the difference between those? Right? They all result in new genetic material being incorporated into the recipient, and that genetic material can contain genes for antibiotic resistance. Okay, so let's get that out of the way up front. Conjugation is basically bacterial mating. What happens in conjugation is that a hollow tube called a pilus is grown from the donor to the recipient, and the genetic material that contains the virulence factor, it's usually on a plasmid, which is a circular piece of DNA that can reproduce independently of the bacterial chromosome. A copy of that is sent to the recipient through the hollow tube. And then once the recipient has the plasmid, they have the virulence factor. And at the end of conjugation, they're both going to have that weapon to produce disease, right? Some of those virulence factors are drug resistance genes. Okay. In transduction, what happens is that the bacteriophage, which is the bacterial counterpart to a virus, right, lands on a bacterial cell, injects its genetic material, which then integrates into the bacterial chromosome. And that integration can bring with it a virulence factor, a weapon, right? And that's called lysogenic conversion. And you can turn a harmless bacteria into a pathogen very easily that way. A lot of pathogens are transduced with virulence factors to cause disease. Okay. Transposition is something that occurs because of mobile genetic elements. Right? Now, what does that mean? There are pieces of DNA that code for enzymes called transposases, whose enzymatic action is to cut that piece of DNA out of wherever it is and then allow it to integrate in another location. Okay. There are transposons in eukaryotes and prokaryotes. Okay. So you name it, there's a transposon around there somewhere. Sometimes these transposons can even grab genetic material from adjacent regions when they deintegrate, right? And then they can insert it into the new target, and then as a result, bring new traits with it. So they are also engines of mutation and also evolution and natural selection. Okay. And then finally, transformation is just the eating of exogenous DNA out there in the environment, right? So there's DNA hanging around the bacterial cell, and the bacteria just eats it, endocytoses it, and then it can express it and even integrate it in some cases. Okay. So that's one of the reasons that antibiotic resistance spreads so rapidly through the bacterial community. Okay. So when you start using a drug, you're eventually going to get to the point where that drug becomes less useful against your target, and then you have to switch. Okay. And so that's one of the things that medical science is constantly doing with antibiotics. All right. Exotoxins are simply enzymes that damage structures in the target so that it can do things like inactivate enzymes critical to protein synthesis, or chew through connective tissue, or whatever. Okay. Weapons. Okay. Secreted weapons. Endotoxins activate inflammation and produce fever. And an example of that we already talked about is lipopolysaccharide. Okay, LPS. Right, LPS. Okay, so it's a structural component in the bacterial cell. Okay. Bacteremia and septicemia are just an indication of bacteria in the blood, right? Bacteria are bacteria in the blood that are not actively growing. Septicemia is the worst possible outcome. Those are bacteria thriving and growing in the blood. And of course, the danger there is it can be spread to every organ in the body and can be lethal. Okay. It's often a result of the failure of our defense mechanisms. Gram negatives are generally responsible. Endotoxins that are released in the blood activate complement and clotting systems and lead to capillary permeability that permits the escape of large volumes of plasma into tissue and contributes to hypotension and, in severe cases, cardiovascular shock. Right. So remember that at any point in time, about 2/3 of your capillary beds are closed up. And that's to help maintain the blood pressure. But when you induce inflammation, you have histamine and cytokine release, and that opens up all your capillary beds at the same time, and that can drop your blood pressure to the point where you can pass out and die. Okay? Septic shock and toxic shock are examples. Right? Viral diseases, right? Most common affliction of humans. Replication depends on the ability to infect the target. It's a non-living pathogen. All right. Why is a virus a non-living pathogen? First of all, it's not cellular. Okay. It's made of a capsid, and that encloses genetic material. Second of all, it can't reproduce on its own. Okay. So it's not a living thing, but it is a pathogen. Okay. So calling it an organism is a little bit of a stretch. Okay. I would call it a simple pathogen. Okay. Viral infections are generally self-limiting. How can they be transmitted? Airborne, in fluid contact, sexual contact, or a vector. Right? Often a vector is an insect, or in some cases an animal. Okay, like a possum or a bat. Right, and it's spread by the bite. Okay, that's how bubonic plague was spread. Okay, wiped out 1/3 of Europe in the Middle Ages, and it was spread by flea bites. Okay. Viral replication happens with the aid of the host. Okay, and what the stages are shown here. Right, attachment, you latch onto your target. Penetration, you get inside your target. Now this can either be receptor-mediated endocytosis, or this could be direct fusion. Okay, depending on whether the virus is enveloped or not. An enveloped virus has host membrane around it that it steals when it leaves the host cell, and a naked virus does not, right? And you have uncoating, right? You remove the capsid components, you disassemble them, you release the genetic material, and then you go into synthesis of genetic material, translation of capsid proteins. Those proteins are assembled into the capsid. That's the box inside of which the genetic material is found. Okay. And then you assemble those and you release them. Now, the release can either be extrusion through the host membrane, so that it, in some cases, steals membrane as it leaves, or it can blow the cell open from the inside out. That's called lysis. Right? So it all depends on what the virus does. Right? The genetic material in a virus can either be RNA or DNA, but never both at the same time. Okay. And the DNA can either be single or double stranded. All right. And the RNA can either be single or double stranded, depending on the virus type. Okay. RNA viruses, with the exception of retroviruses, do all of their replication in the cytoplasm. Right. Whereas DNA viruses use the nucleus of the host and are much more difficult to get rid of. Okay? And in some cases, they stay inside you your whole life, such as herpes zoster, which produces shingles and chickenpox. Right? Viral replication, we make copies of genetic material. The new virions are released from the cell to infect other targets. Some remain latent until they're activated by we really don't know, right? Lots of different things, right? Stress, hormone changes, other diseases. Okay. The entire herpes class likes to integrate and sleep inside your genetic material, usually in nerve cells. Okay. And then it erupts from there at a later point if activated. Okay. And you can never get rid of it, unfortunately, right? There's no way to go into every cell that's been compromised and remove that information and then restore normal function, at least not yet. Okay. Um, because of this, viruses are mutagens, right? They mutate the cells that they attack if they're DNA viruses, and that can lead to things like cancer. Okay. Um, latency is also a mechanism used by microbes to avoid being destroyed. It's a way to go to sleep, right? We have to appreciate the fact that when we're talking about viruses, okay, and other microbes, right? There's usually a stage where we just can take a nap. Okay, for viruses, it's a latency where they integrate into the host and just wait to be activated again. In bacteria, it's the endospore, all right, which is a version of the bacterial cell that can withstand very poor environmental conditions and will not go into vegetative growth until good conditions arise later on. In fungi, it's the spore, okay, which can resist, again, uh, poor growing conditions, right? And then finally, in helminths and protozoans, it's the cyst. Okay? So all of these are stages in the life cycle that are kind of like immortality, right? We can exist like this almost indefinitely until the appropriate conditions come around again. Okay. As an example, right? They found endospores in the ice cores that they've taken out of the Arctic that date back to the Jurassic period, which is when dinosaurs ruled the earth, and they've managed to get them to grow. Okay. So that's millions and millions of years, right? That's pretty impressive. Okay. So there you go. The secret to immortality right there in the humble endospore, right? So examples in the plant, it's the seed, right? Seed can last almost indefinitely. Okay? What do viruses do that causes damage? How is it that they hurt us? Well, they inhibit host cell DNA, RNA, and protein synthesis, and so they take the cell off task. When you take the cell off task, it's not doing its job, and that's what can produce disease, right? We can disrupt lysosomal membranes and release enzymes that damage the host. Remember, the lysosome is the stomach of the cell, and those lysosomal enzymes can basically tear up any protein they encounter, right? Transformation of the host cell into a cancer cell, because again, the virus is a mutagen, all right? It can mutate cell cycle genes. Promotion of secondary bacterial infection. Why? Because you've weakened now some immune function in many cases, right? And a glaring example would be AIDS, right? Where you infect the helper T cell and you suppress adaptive immunity in both the humoral and the cell mediated branches. Okay. And then you can have fusion of infected cells to produce what are called syncytia. Let's write that down. Okay. Giant fused cells that don't function properly. Okay. Changing antigenic properties of the host can lead to the immune system to attack the cell as if it were foreign. Right? There are some bacteria that do that too. Right? Antigenic variation is where we can change our surface appearance, and that can spread our host range, but can also make it more difficult for the immune system to find us and clear us. Okay. Antigens responsible for protection against flu undergo annual change, right? Minor change we see is called antigenic drift. Major change is antigenic shift. Now, remember that since a virus is not a living thing. It can tolerate more mutation than a living pathogen can because it doesn't really do metabolism. Okay. So if the changes happen in coat proteins, then it can avoid an immune system that was prompted to respond to a previous strain, right? Very tricky. Another thing you can see is that you can sometimes form hybrid viruses. The figure is showing you what happens, for instance, when two viruses co-infect the same host, and then a hybrid gets produced that can now infect a host that it couldn't infect before, right? So this is how avian flu can get to humans, H1N1, right? We have a human virus, and then we have an avian virus that co-infect a common host, the pig. They form a hybrid, right? That hybrid bears genetic material and capsid proteins from each, and now the hybrid can hit either the bird or the human. Okay, another nasty trick that viruses can pull. Okay, now fungi are different. Okay, fungi are eukaryotic microbes that feed off of dead organic material. All right, they have rigid cell walls made of chitin. Okay, um, they exist either as single-celled or multicellular organisms, and sometimes they can exist as either or, right? They reproduce by simple division or budding, but they can also engage in sexual reproduction, right? Depending on the type of fungus we're talking about. Diseases that are caused by fungi are called mycoses. They can be superficial, meaning they're on the skin or the mucous membranes, or they can be deep, or they can be opportunistic, meaning they take advantage of a weakened immune system. Fungi that invade the skin, the hair, or the nails are called dermatophytes. These diseases are called tinnias. Okay, example: tinea capitis, cradle cap; tinea pedis, athlete's foot; tinea crurus, jock itch. Okay, um, the pathogenicity is the ability to adapt to the environment. Right. Remember that on your body you have a lot of different little microenvironments, temperature variations, moisture variations. Okay, um, they can adapt to these. They can digest keratin as a fuel source, and they can even exist in low oxygen conditions. Okay, they can grow anaerobically in many cases. They can suppress immune defense, and they're usually kept in control by phagocytes and T lymphocytes. Okay, so a perfect example of a fungal infection would be candida. Okay, candida albicans, alba means white. So when this grows, it makes a white filamentous coating on whatever it's growing on. Okay, it's an opportunistic infection. It's found in the normal microbiome of skin, GI, vagina. Localized infection of, if overgrowth occurs, is usually seen in immunocompromised individuals. Okay, which may result in deep infection and have a high mortality rate. So this would be somebody on chemo, radiation, or somebody with AIDS. A condition that you might recognize that's a candida opportunistic is thrush. Okay. Okay. When somebody with thrush opens their mouth and says, their oral cavity is coated with white fuzz. Okay. And that's the candida, right? So, where'd the candida come from? Well, it's everywhere. It's on you, in you, and around you right now. Okay. But you keep it in control because your immune system is able to deal with it. Okay. Parasitic infections. The parasite benefits from the host. Okay, the host provides the parasite necessary components to live, gives it a home, it gives it nutrients. Okay, parasites can be protozoans or helminths. Okay, protozoa are single-celled eukaryotes, and helminths are multicellular eukaryotes. Okay, such as flukes and nematodes and tapeworms. Protozoa include the malarial parasite, okay, amoeba and flagellates, all right, single-celled organisms, right? Um, they're more common in developing countries because of the lack of public sanitation in those regions where the potable water and the wastewater are often the same, right? The potable water is the drinking water, okay? So they can be spread by fecal oral, they can be spread by insect. It all depends on what sort of parasite we're talking about. They can be ingested, right? Tissue damage is secondary to infestation with toxin damage from in the inflammatory response that takes place. Okay. So how do we deal with this, right? Reemergence of some diseases due to lack of implementation or breakdown in application. Environmental measures include waste disposal, water treatment and contamination prevention, food safety, vector control, okay? Insecticides and antimicrobials can be used, right? Insecticides to control the vector, right, which is the agent that carries the pathogen from one location to another. Otherwise, it wouldn't be able to spread as easily, right? Bactericidals and bacteriostatics, they have different modes of action, right? Bactericidal means it kills the bacteria. Bacteriostatic means it keeps the bacteria from growing, okay? So we can inhibit cell wall synthesis, we can damage the plasma membrane, we can change the metabolism of nucleic acids, we can attack the ribosomes of the microbe. For instance, the prokaryotic ribosome is functionally identical but structurally different from the eukaryotic ribosome. So we can design drugs that go after that and leave our own ribosomes alone. Okay. And we can modify energy metabolism in the microbe. Okay. Antibiotic resistance results from genetic mutation, followed by evolution and natural selection. Inactivation of the antibiotic is seen in things like penicillin resistance, where we basically cleave portions of the antibiotic that normally produce the toxic effect. We can have modification of the target molecule, okay? We can increase the active efflux of the antibiotic. It's often caused by lack of compliance with therapeutic regimens that allow selective resurgence and overuse of an antibiotic that destroys your native flora and allows the pathogen to take off. Okay. So this is one of the reasons this selective resurgence is one of the reasons that doctors will shift from second generation to first generation antibiotics. What you do is you change your counter weapon and you can wipe out something that's resistant to, say, a certain class of antibiotic with a different class of antibiotic. And then you do that until that antibiotic becomes less effective and then you can switch back. Okay. So it's a game you play, right? Vaccines are just preps of an antigen meant to induce a primary immune response in the person that receives the injection. Okay. And so what happens is that the person that designs the vaccine selects antigens and puts them in the shot. You get the injection of those antigens and you generate a primary immune response, which makes memory cells against the antigens that are in the vaccine. Okay. And then when you encounter that antigen a second time on a pathogen, you're not going to get sick because you have those memory cells standing by waiting to do their thing. Okay. They can provide long lasting immunity. Okay. But their development is expensive. Some people are reluctant to vaccinate, but complications from vaccination are rare. They can induce long-lasting protective immune responses that will not result in disease in a healthy recipient, okay? And they can also serve as a way to knock down toxins, okay? To cloak them so that they don't produce their effect. Now, that said, all right, when vaccines fail, why do they fail? All right. They fail either because they produce an adverse event in the person that receives the vaccine, okay, in a high enough percentage of the recipients to not warrant the risk, okay, or they fail because the target of the vaccine, right, the pathogen you're going after has a high mutation rate. If the pathogen has a high mutation rate, then the vaccine becomes useless, okay? Because you'll end up generating a primary immune response to what's in the vaccine, but because the pathogen mutates at such a high rate, the likelihood that you're going to pick up the disease again is very high, okay? So we can give a couple of examples of a successful and an unsuccessful vaccine. Okay. Um, a successful vaccine would be the polio vaccine, which was actually developed at UC um, back in the 40s. Right. And it was very effective. Okay. Um, and has been effective for decades. And that's because the polio virus, right, mutates at a low rate. An example of an unsuccessful vaccine, unfortunately, are the COVID-19 vaccines. Right, COVID belongs to a class of viruses that we generically call cold viruses. Okay, cold viruses mutate at a very high rate. All right, that's just their nature. So when you generate a vaccine against a cold virus, you'll be resistant to what's in the vaccine, but the likelihood that you're going to run into a strain that's different is very, very high. Okay. And you can see with COVID that we're on, I don't know, the 25th iteration of the vaccine now. All right. When you have a condition like that that's spread by something that mutates at a high rate, the most effective immunity is called natural active immunity, which is basically when you get sick and then get better because of the action of your immune system. And the reason that natural active immunity is superior to artificial active immunity vaccination is because when you acquire a pathogen, your immune system is going to develop antibodies and cytotoxic T's against every single foreign antigen on that invader, not just one selected by somebody that designs a vaccine. Okay. And as a result, you're going to have a much more robust immunity to that organism the next time it shows up. Okay. Um, Another thing that happens when a disease runs its course through a population is it's going to select for the individuals that are resistant to it. And once that happens, those are going to become the predominant individuals in the population, and susceptible individuals will be protected because they're basically cloaked by a blanket of resistant individuals. And so the disease is going to have a difficult time reaching them. Right? It's tough for it to spread, and that's called herd immunity. Right? And that's essentially where we've gotten to now with COVID. Right? All right. Common problems, right? It's tough to access the vaccine in developed countries. Lack of compliance is a big one, right? Unresponsiveness to the vaccine and resistance to the vaccine, right? And then reluctance to take it, okay? Passive immunotherapy is a little bit different. You're getting antibody from another source. Okay? Natural passive immunity happens through cord blood and mother's milk, right? You get antibodies from your mom while your own immune system is being set up, right? Whereas artificial passive immunity, you're getting an injection of antibody through a needle, right? And that is um, all passive immunity is temporary, right? It only lasts as long as the antibody's in your system. Once the antibody clears, you're vulnerable again, okay? So examples, right? Human immunoglobulins for hepatitis, immunoglobulin and monoclonal antibodies for rabies, monoclonal antibody for RSV, okay, are some examples, right? So you might wonder, well, when would I, you know, when would I go and get a artificial passive immunity shot, right? Well, early stages of a rabies infection, like you have a bite, right? You want to go in and get the shot, which includes the antibody and the toxoid, okay? Um, but another example would be if you're going to travel to an area where a certain disease is endemic, okay? Such as if you're going to equatorial regions of the planet where malaria is an endemic disease, you might get a shot of anti-malarial antibody to keep you safe for a couple of weeks. Okay. Immune deficiencies are they're going to fall into one of the three categories we talked about, right? Hypersensitivity, immune deficiency, or autoimmunity, right? So these are failures of self-defense, okay? Primary immunodeficiency is usually inherited, okay? So there's a busted gene somewhere that doesn't let you make either all of your immune system or components of your immune system. Secondary immunodeficiencies are caused by either a treatment like chemo and radiation or acquired by infection of something like AIDS. Okay. These are more common, all right? Clinical presentation, development of recurrent severe infection. T cell deficiencies are usually the result of viral, fungal, yeast, and atypical microbe activity, whereas B cell and phagocyte deficiency are microbes that require opsonization. Okay. You can also have complement deficiencies, right? That comes from liver damage. Talk about the fact that the liver makes clotting factors and complement, okay, as well as things like albumin that helps you hold on to your blood volume. And when you have liver disease, you don't make the complement, you don't make the clotting factors, and you get infections more easily, while you also bruise and bleed more easily, okay? Primary immune deficiencies are usually single gene defects, right? They're generally, they can be inherited or they may not be inherited. It all depends, okay, when the mutation takes place. They can appear early or late in life. They're rare, but they are increasing. Major groups include B and T lymphocyte deficient individuals, antibody deficient individuals, immune dysregulation, phagocyte defects, innate immunity defects, and complement defects. Okay. When we come back from break, we'll talk a little bit more about this. Okay. So let's take 15 and we'll come back from break.
11.2 deletion syndrome is a syndrome which is caused by the deletion of the small segment of chromosome 22. So in the chromosome 22, a very small portion in the Q arm is deleted and the locus is known as 22q11.2. And as a result of this deletion, there is DiGeorge syndrome. So simply, DiGeorge syndrome is an inherited immunodeficiency disorder. We would learn about it in details. But the major facts about this disease is they have poorly developed thymus or no thymus at all. So you can clearly understand that there would be effects in T cell production and these individuals would be immunocompromised. Lastly, these individuals undergo cardiac complications. So let us look at some facial features that are distinct to enlarged nose tip and a hooded eye, and majorly they have a very long face. So these are some few characteristics, facial feature of DiGeorge syndrome. Now this disorder is typically due to a deletion. Now due to this 22q11q2 deletion, there is at least 20 genes or more than 20 genes are deleted or affected. One of them, or one of the most important of them, is TBX1 gene. TBX1 gene is an important transcription factor which is present in the third and fourth pharyngeal pouch. And also it is enriched in facial structures, heart, thyroid, parathyroid, thymus, etc. And they're present while these tissues are developing. So in DiGeorge syndrome, TBX1 gene is missing. And as a result, these structures are compromised. So we can clearly understand that TBX1 is highly expressed in the structures such as heart, thyroid, parathyroid, etc. And all of these structures would be somehow perturbed in a DGS patient. Majorly, the heart is affected. So this is how a normal heart looks like. But in the DGS patient, you would see ventricular septal defect or atrioventricular canal defect. These kind of defects are pretty common in DiGeorge syndrome patients. Other than heart, there would be complications associated with thyroid and parathyroid gland. Now, let me tell you another important feature of DiGeorge syndrome, which is presence of a cleft palate. So here, a opening or cleft in the roof of the mouth plate is seen. So you can see the upper lips are not totally joined to each other. Now, as a result of this, there could be difficulties in swallowing, and there could be also speech-related or verbal difficulties. Characteristic sound might come out. So these kind of differences are present in DiGeorge syndrome patients. Now, although most of the DiGeorge syndrome patients suffer to some extent immunodeficiency, but the extent of immunodeficiency could be variable. Now, the most important thing is thymus is not present or partially present in DiGeorge syndrome patients. So you can clearly understand T cells which develop from the thymus is not present in case of these DiGeorge syndrome, or even if present at a very small extent. So all the T cell mediated functions of the immune systems are abrogated, and these patients are more vulnerable towards opportunistic pathogen, bacteria, etc. Now, thymic transplantation and passive antibody treatment might be beneficial for these patients, but this is not a long-term solution. So these individuals have their cell-mediated immunity severely compromised, as well as the B-cell-mediated humoral immunity is also compromised because B-cell activation requires T-cell. So that is why these individuals are vulnerable to bacterial and fungal infection. So I hope that summarizes a little bit about DiGeorge syndrome, and this is a very short video, but if you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you. And if you want to.
So with the DiGeorge syndrome, what you're getting is no T cell production. And since the helper T cell stimulates both cell-mediated and humoral immunity, then you don't really have an effective adaptive immune response, okay? Antibody deficiencies can involve defective B cell development and may only hit one class of antibody or several. Common immune deficiencies include hypogammaglobulinemia and agammaglobulinemia, okay? Basically, what's happening here is that certain classes of antibody are not made. Remember that there are several classes of antibody: IgG, IgM, IgD, IgE, okay, G and IgA, right? And they all operate in different parts of the body. But the antigen binding specificities, right, are basically slapped on to those constant regions by the same plasma cell. And they just define where the antibody is going to do its job. So with these type of conditions, you're missing one or other class of antibody, right? Phagocyte defects include chronic granulomatous disease, which is a defect in the myeloperoxidase hydrogen peroxide system, okay? It's a genetic disorder in which white blood cells called phagocytes are unable to kill certain types of bacteria and fungi. People that have CGD are highly susceptible to frequent and sometimes life-threatening bacterial and fungal infections. It's caused by a defect in NADPH oxidase that phagocytes need to kill certain bacteria and fungi. Mutations in one of the 5 different genes that produce this can cause these defects, okay? So we see recurrent pneumonia, tumor-like granuloma in lungs, skin, bone, and other infection. Defects in innate immunity, okay, our granulocyte activity and our inflammatory response include things like chronic mucocutaneous candidiasis. That's a manifestation of this condition. Recurrent candida infections due to defective immune responses to candida, okay? So you would see things like thrush, all right? Complement deficiencies, okay? C3 deficiency is the most severe defect due to the central role in the complement cascade. Remember that complement complements the action of antibodies so that they're more effective at clearing the infection, Okay, and they're all made by the liver. Okay, this results in recurrent life-threatening infections. Okay, mannose binding lectin deficiency is a condition that affects the immune system. It's fairly common, affecting approximately up to 30 people in every 100, so up to 30%. People with this condition have low levels of an immune system protein called mannose binding lectin in their blood. Genetic changes in the MBL2 gene can lead to this deficiency. This gene provides instructions for making MBL protein that plays an important role in the body's immune response. MBL protein attaches to foreign invaders like bacteria, viruses, or yeast, and activates other important parts of the immune system known as complement. Okay, so without that, we don't get the lectin pathway. Okay. Secondary deficiencies, also referred to as acquired deficiencies, are more common than primary deficiencies, and they're often not clinically relevant, but they are in certain special cases, right? When we look at somebody's immune function, okay, we need to do a CBC in order to determine whether they have the requisite number of white blood cells in each class, okay? So we ask the question, what's your eosinophil count? What's your basophil count? What's your neutrophil count? What are your lymphocyte counts and macrophage and monocyte counts? Okay. We can also do a quantitative determination of immunoglobulins, okay, an assay for total complement in order to see how your function is, okay. With immunodeficiency, we can provide gamma globulin, which is IV immune globulin. This is, again, passive immunity, artificial passive immunity. We can do stem cell transplant, which means that we're going to swap out your marrow, okay, or a component of your marrow to try to restore what's missing. We can transfuse erythrocytes. We can do mesenchymal stem cell injection. And then way out there on the edge is gene therapy, where we replace defective genes with functional ones. Now, one of the examples of acquired immunodeficiency is AIDS. Let's take a look at how AIDS works.
HIV is the human immunodeficiency virus. If you have HIV, you have an infection that damages your immune system over time and causes AIDS. AIDS stands for Acquired Immunodeficiency Syndrome. It is the final stage of an HIV infection when your immune system is damaged and too weak to fight off ordinary infections. When foreign invaders, such as bacteria and viruses, get into your body, they can cause infections. These events activate your body's defenses. The white blood cells of your immune system are part of your body's defenses. One type of white blood cell, called helper T lymphocytes or helper T cells, strengthen your immune system's response to infection in two ways. First, helper T cells release chemicals that attract other white blood cells to the site of the infection. These additional white blood cells attack the invading bacteria or virus, as well as other infected cells. Second, helper T cells release chemicals that cause other white blood cells to multiply. These new white blood cells create markers called antibodies, which can identify the same foreign invader throughout your body. Antibodies attach to the bacteria or virus, marking them as targets for your immune system to destroy them. If you have HIV, it travels through your blood and other body fluids to infect and kill certain white blood cells. The virus enters helper T cells, which are the primary target. Once inside, the virus makes many copies of itself. As these virus particles are made, They leave the damaged helper T cell to infect other cells. The T cell loses its ability to protect the body from the ongoing infection and dies. In this way, HIV spreads and kills more of your helper T cells, weakening your immune system. As a result, other types of infections are able to take advantage of your body's inability to defend itself. These infections are called opportunistic infections. If you have an HIV infection and one or more opportunistic infections, you have AIDS. Some of the common AIDS-related opportunistic infections are inflammation of the tissues covering your brain and spinal cord, called meningitis, inflammation of your brain, called encephalitis, respiratory illnesses, such as pneumonia and tuberculosis, intestinal illnesses, such as chronic diarrhea caused by infectious parasites, and cancers, such as Kaposi's sarcoma and non-Hodgkin lymphoma. HIV passes from person to person through infected body fluids. HIV can enter your body during unprotected sex, while sharing drug injection needles, during your own childbirth, while breastfeeding from your mother, or from contaminated blood or blood products. Although there is no cure for HIV, drugs called antiretroviral medications can reduce the amount of HIV in your body. One class of antiretroviral medication, called entry or fusion inhibitors, disrupts the HIV infection process by preventing the virus from attaching to your cells. Other classes of antiretroviral medications include reverse transcriptase inhibitors, protease inhibitors, and integrase inhibitors. These drugs prevent the creation, assembly, and spread of new viruses. Your doctor may prescribe a combination of these drug classes known as highly active antiretroviral therapy, or HAART. Antiretroviral medication doesn't completely remove HIV from your body, but slows it down enough to enable your immune system to fight infections. Regular blood tests will let your doctor know how effective your antiretroviral medication is in controlling HIV. If the number of helper T cells is high enough in your blood sample, your medication is working. Treatments for the opportunistic infections of AIDS are medications specific for each type of infection. For example, your doctor may prescribe antibiotics if you have pneumonia or tuberculosis. To avoid getting or spreading an HIV infection, know your HIV status and your partner's status by getting tested regularly. The most effective way to prevent HIV infection is to avoid vaginal and anal sex. When engaging in sexual activity, you will be less likely to contract HIV if you only have sex with one uninfected partner or use latex condoms for protection. Avoid using injectable illegal drugs or sharing drug needles because the needles may have the virus on them. Avoid intoxication from drugs or alcohol because you will be more likely to engage in unsafe sexual behavior.
So the thing to realize about AIDS is that it's a viral infection that helper T's that takes them off task, right? And it turns them into virus factors. In addition to that, it also causes the helper T's to express something called fast ligand, which when it hits anything with fast receptor on it, it kills the cell. So it turns it into a suicide pill that kills anything it comes into contact with. And so your white count drops dramatically as a result of this. Okay. In addition, all right, because the helper T population is basically wiped out, you have no adaptive immune response whatsoever. All right. The helper T normally stimulates both humoral and cell-mediated immunity. With an absent helper T population or dropping white count, opportunistic infection and more frequent instances of cancer come up because the natural killers are not properly stimulated as well. Okay. And the result then is you're going to get all of these opportunistic infections and conditions setting in, right? Pediatric AIDS is transmitted during pregnancy, at delivery, or through breastfeeding. It's more aggressive in children. If untreated, the child will likely die by a second birthday, and there's often neurologic involvement, okay? HIV-induced encephalopathy may be difficult to differentiate from other risk factors. Basically, when we're talking about encephalopathy, we're talking about inflammation of the brain, all right? And of course, the problem here is that neurons are destroyed and they do not grow back, right? Hypersensitivity is altered immune response to antigen that results in disease or damage to the host, okay? Basically, your immune system overdoes it, all right? Allergy is the damaging effects of hypersensitivity to exogenous antigens, right? Autoimmunity is the direction of your immune system against your own antigen. And alloimmunity is an immune reaction to tissues from another individual. And this is expected, okay? If you don't have a tissue match for something like a transplanted organ or a blood transfusion, you're going to destroy the incoming tissue as alien tissue, right? Because remember that the immune system bases its attack on self versus non-self and not good versus bad, okay? So even though that tissue is intended to help you, it's marked as alien and will be wiped out, right? So we characterize this by a variety of different types of hypersensitivities, types 1 through 4, all right? Let's take a look at how they do what they do.
People develop an allergic reaction or hypersensitivity when exposed to substances such as dust, pollens, animal dander, or penicillin. Sensitization occurs when the antigen makes contact with some part of the body. The antigen is taken up, processed by antigen-presenting cells, and presented to T helper cells. Under the mucous membranes are rich in B cells committed to IgE production, and IgE-producing cells are more abundant in persons susceptible to allergies. The T helper cells produce cytokines, which stimulate B cells in the area to proliferate and differentiate into IgE-producing plasma cells. IgE is produced in specific areas of the body. The IgE molecules attach to nearby mast cells. The individual is now sensitized to the antigen. When exposed to the antigen for a second time, the antigen immediately binds to the IgE antibodies, which are already attached to the mast cell. Within seconds of this binding, the mast cell releases histamine and other chemicals that cause an inflammatory response. These chemicals trigger a variety of symptoms such as capillary dilation, airway constriction, mucus secretion, pain, and itching. Two hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. Leads to complement fixation and lysis of these cells. Blood from a person who is type A is transfused into a person who is type B. Antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. Also leads to complement fixation and cell lysis. Patients who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients. Immune complexes consist of antigens and antibodies bound together. Although large immune complexes are engulfed by phagocytes and removed, intermediate-sized immune complexes are not readily removed. These intermediate complexes react with and activate complement. For example, complement activation in blood vessels causes basophils to degranulate, which causes vasodilation. Immune complexes entrapped in blood vessels or other tissues, such as kidney glomeruli, activate complement, which attracts neutrophils and causes them to degranulate. Neutrophils release enzymes responsible for tissue damage. Effects produced by the mechanisms of cell-mediated immunity are referred to as delayed hypersensitivity. Delayed hypersensitivity is mediated by Th1 cells, also called TD cells, and is responsible for contact dermatitis, tissue damage in a variety of infectious diseases, and rejection of tissue grafts. Contact hypersensitivity, such as poison oak dermatitis, is initiated by contact with the antigen, which is also called the allergen. Contact, a small molecule from the plant, called a hapten, binds to a carrier protein in the host. Carrier protein with bound hapten is ingested by a macrophage, and the hapten peptide is then presented on the surface of the macrophage on a class II MHC. Th1 cells with T cell receptors capable of recognizing the hapten peptide antigen interact with the presented antigen, become activated, and increase in numbers. Host comes in contact with the allergen a second time. The Th1 cells react with the hapten peptide being presented on the macrophage and release cytokines, resulting in attraction of more macrophages, followed by inflammation and skin lesions. Characteristic skin lesions appear after 24 hours, reaching their peak at 48 to 72 hours after the second exposure to the plant.
So 4 different classes of hypersensitivity. Now, how to deal with them? You have to use anti-inflammatories and immune suppressants, right? Allergy is the most common hypersensitivity, and usually type 1. Environmental antigens that cause atypical immunological responses in genetically predisposed individuals include pollens, mold, fungi. It can be anything, really. Often the allergen is contained within a particle too large to be phagocytized or is protected by a non-allergenic coat. Okay. Anaphylaxis is the most negative outcome of hypersensitivity. Okay. Rapid and severe hypersensitivity occurs within minutes of re-exposure to antigen. It can be systemic or cutaneous. Most severe reactions can lead to shock and death. And examples include bee stings, peanuts, shellfish, or eggs. There's others. Okay. How do we deal with this? Well, one way is desensitization to reduce the severity of the allergic reaction. But it could also cause anaphylaxis. We really don't know. So the idea with some of these, you probably heard of allergy shots, right? And the idea with allergy shots is you inject the allergen into the bloodstream. You have to kind of hunt for what the allergen is. Okay, so you use various mixtures of allergens, and then when you get something that produces a reaction, then it's in that mixture somewhere, and then you can continue to break the mixture down until you finally find what it is you're allergic to. But once you do, you can inject small amounts of it into the blood and generate a primary immune response using IgM. And the IgM is thought to intercept the allergen when you encounter it and not let it get to the IgE, okay, which is what's going to trigger the histamine and cytokine release when it jumps on the mast cells. And then you can basically work your way out of the hypersensitivity. It doesn't always work, okay? It doesn't always work. The other way, of course, is to take antihistamines and anti-inflammatories, and that reduces the problem with the allergy, but it leaves you open for infection, okay? With autoimmunity, what's going on is that we're turning around and we're attacking our own tissues. And we talked at the beginning of the lecture about why we think this happens, all right? There's a variety of theories out there. It could be one or the other, or it could be a combination, okay? But it's a breakdown of tolerating our own antigens, and can lead to infectious disease, okay? For instance, rheumatic fever can lead to heart problems, okay? Glomerulonephritis can be the result of an autoimmune attack on the kidneys, all right? Graves' disease is an autoimmune attack on the thyroid, and type 1 diabetes is an autoimmune attack on the beta islets of the pancreas, okay? So, you know, one of the most glaring examples of autoimmunity is systemic lupus, okay? It's a multi-system inflammatory disease where you generate autoantibodies against all of the stuff listed below, okay? And it has system-wide effects. So let's take a listen to systemic lupus. I think this is the oh,
Lupus is a disease in which your immune system attacks the tissues in your own body. The immune system is made up of white blood cells that defend your body against foreign invaders. Normally, when foreign invaders, such as viruses and bacteria, get into your body, some of your white blood cells identify them as foreign because they contain substances called antigens. Recognition causes some of your white blood cells to make chemical markers called antibodies. Antibodies attach to the antigens, marking the foreign invaders as targets for your immune system to destroy them. An autoimmune disease, such as lupus, your white blood cells mistakenly make antibodies to mark some of your own body's cells and circulating chemicals for destruction. Antibodies attach to your normal cells. White blood cells begin to attack them as if they were foreign antigens. Time, this autoimmune response causes inflammation in the parts of your body wherever this attack is happening. Inflammation makes the blood vessels in the affected area expand and leak fluid, causing redness and swelling. Symptoms of lupus happen as your immune system begins to attack different areas of your body. You may develop a butterfly-shaped skin rash on your cheeks and nose. Sores or ulcers may form inside your nose or mouth. You may develop arthritis, in which your joints become inflamed and swollen. If fingers are cold, they may turn a blue or white color. This is called Raynaud's phenomenon. You may have pain during deep breathing caused by pleurisy, a condition in which the outer lining of your lungs is inflamed. White blood cells may create antibodies that attach to bits of your DNA from worn out cells. The attached pair, called an immune complex, can lodge in your kidneys and cause inflammation. This disorder is called lupus nephritis. Flare-ups of any of your symptoms, even those not related to your skin, may be caused by an unusual reaction to sunlight called photosensitivity. Common symptoms of lupus include extreme fatigue, aches, fever, reduced number of red blood cells in your blood called anemia, swelling of your legs, feet, hands, or skin around your eyes, pain and weakness, hair loss. This is sometimes called the great imitator because its many symptoms are found in other health problems. No cure for lupus. However, you may prevent or reduce flare-ups by avoiding direct sunlight and wearing sunscreen outside. Sunscreen should have a sun protection factor of 55 or greater and block both ultraviolet A and ultraviolet B radiation. To treat your symptoms, your doctor may recommend a combination of drugs to reduce inflammation, such as non-steroidal anti-inflammatory drugs, or NSAIDs, anti-malarial drugs. Your doctor may also recommend drugs that suppress or prevent your immune system from attacking the tissues in your body, such as corticosteroids.
With alloimmunity, what you're looking at is a predicted immune response to variations in an antigen within a species. Okay, an example is the type 2 hypersensitivity that we see in the ABO blood groups, right? Antigens A and B are carbohydrates on the surfaces of erythrocytes, and essentially the O is a carbohydrate assembly against which there is no human antibody. Okay, so the different blood types listed, right, A, B, O, and AB, are inherited in such a way that let's get the pen going here. Okay. The alleles are IA, IB, and little I, right? Little I is for O, IA is for A, and IB is for B, okay? So the way it works is that IA and IB are what's called codominant to each other, meaning if you have one of each, you're going to have AB, okay? Right? O is recessive to both A and B, all right? So the only way you get O is to have two O alleles, one from mom and one from dad, all right? Type A, you can get it two ways, right? You can have two IA alleles, which means you're homozygous for the A gene, IA, right? Or you can be IA little I, right? That's also type A. Same thing with B. You can be IB little I, which is type B, or you can be IB. And those will both give type B blood, right? Now, the way that the antibody distribution works is that if you have a type A individual, you're going to have type B antibody. If you're a type B individual, you're going to have type A antibody. If you're an AB individual, you're going to have neither antibody. And if you're an O individual, you'll have both type A and type B antibodies. So the way that this works is that you're never going to make antibodies against your own antigens because you would then agglutinate your own blood, your red blood cells would burst and you would die. All right. So when we transfuse, right, we worry about the antibodies of the recipient and the antigens of the donor blood, right? So this is why people who are type O are called universal donor, right? They can give to any blood type. I'm a type O, so I often get calls from areas where you could donate blood, like Hawksworth and up here in Dayton, Red Cross and places like that. And that blood can be used in any transfusion. Okay. I'm also Rh negative. All right. That's another antigen we'll talk about in a minute. If you're type A, you can donate to A or AB because people who are AB don't have antibody against those antigens. If you're type B, you can donate to either B or AB. Okay. And if you're type AB, you can donate to AB. All right. That's it. That's the way it works. So AB is what's called universal recipient, and O is called universal donor. All right. That's the way it works. Right. Now, when we're talking about the Rh group, all right, you're either Rh negative or Rh positive. Okay. Okay. It's a dominant allele. All right. And the name of the gene is D. Okay. You either have the capital D allele, which is the Rh positive, or you have the little d allele, which is Rh negative. Right. So if you're big D, little d, you're Rh positive. Okay. If you're big D, you're Rh positive. Okay. And if you're little d, you're Rh negative. Now, your Rh status and your ABO status have nothing to do with each other. Okay. They are separate genes inherited separately. All right. So, for instance, if we say that somebody is O negative, okay, they have the O antigen and they do not have the Rh antigen. If they are AB negative, they have the A antigen and the B antigen, and they don't have the Rh antigen. If they're AB positive, all right, they have all three antigens. They have the A antigen, the B antigen, and the Rh antigen. Right? Rh positive individuals will never have antibody against Rh for the same reason we talked about with the ABO groups. They would agglutinate their own red blood cells, they would rupture, release hemoglobin that would block capillary beds, and you would die. Okay. But an Rh negative individual will have Rh antibody if they've been exposed to it at some point. All right. So that happens either in a mismatched blood transfusion or it happens in an Rh negative mother if she has an Rh positive child. Okay. Because at birth, when the placenta is torn from the uterus, there's a mixture of the child's blood and the mother's blood, and then she's going to generate Rh antibody at that point. If she has a second Rh positive child, her antibodies can cross the placenta and get into the baby's blood and rupture the baby's red blood cells and potentially kill the child. So one of the things that we do to deal with that is we give you a drug called Rogam, R-H-O-GAM. Okay. And what it does is it cloaks the Rh antigen. And as a result, a mother can have as many Rh positive children as she wants if she's Rh negative, because we give the drug before, during, and after the pregnancy. And so even when the baby's born, she's going to have enough of that Rogam in her system that she can block that antigen from her immune system, and she'll never generate antibody against the Rh antigen. Before the advent of Rogam, you had to have blood tests to determine your compatibility. And if the man was Rh positive and the woman was Rh negative, you had a 50% chance on each pregnancy of having an Rh positive baby. All right. Now, the first pregnancy will go fine. All right. But it's the second pregnancy that's in danger. So before Rogam, you might have just one child if you were incompatible, or no children, right? Or maybe you would adopt, or maybe you wouldn't get married, right? But now with Rogam, we don't have to worry about that, all right? You just give it and then block the antigen and you don't make the antibody to it. Now, another important point here is that if both mother and father are Rh negative, you don't need to do this because you can't make an Rh positive child. It's not possible. You don't have the alleles to do it, right? Or if the mother is Rh positive, right? Because she's never going to make antibody against her own antigens, right? So if mom is Rh positive, or if mom and dad are both Rh negative, we don't have to worry. It's only when mom's Rh negative and dad's Rh positive that it becomes an issue. Okay.
What time is the quiz?
Well, say again.
The quiz, what time is it?
Oh, we're getting ready. This is the last slide. Transplant rejection is classed according to time. Okay. Hyperacute is usually immediate and rare with pre-existing antibody to the antigens in the graft. Acute is cell-mediated immunity against unmatched HLA antigen. And again, we would expect this to happen, all right? And chronic is months or years and the result of a weak cell-mediated reaction against minor HLA antigens. So generally speaking, all right, when we do transplants, we want the HLA antigens to be 85% or better homologous, meaning similar in amino acid sequence before we even attempt it. And even then we have to use immune suppressants to try and get the desired outcome. Okay. All right. With that, we are done.